Hello. We're continuing with um, life science and cell biology in a nutshell. So, we are learning currently about the um, DNA, and we were learning that um, chromatin is the denser form of DNA inside the nucleus of cells. When the cell is in interphase, or the stage of the resting stage where it's not undergoing replication. So, there are two types of chromatin. Euchromatin is the genetically active portion, is involved in transcribing RNA to produce proteins in cell function and growth. So when transcription occurs, RNA is made off of the DNA template. Then the RNA goes and proteins are made out of the off the RNA strand. This occurs in the ribosomes. Heterochromatin contains inactive DNA. By contrast, is the portion of chromatin that is most condensed, and it is not. It is the state that the chromatin is in when the DNA is not accessed or it's not used. Throughout the life of a cell, chromatin fibers take on the different forms. During interphase, when the cell is carrying out normal functions, chromatin is dispersed throughout the nucleus in what appears to be a tangle of fibers. This exposes the euchromatin and makes it available for transcription. So when it's loose, it's euchromatin. When it's uh, off of inaccessible, it's heterochromatin. Outer membrane, inner membrane, nucleolus, nucleoplasm, and the chromatins, the heterochromatin, the inactive, and the euchromatin, the active. More, then the ribosomes towards the outside, the nuclear pores. When the cell is entering metaphase and preparing to divide, the chromatin changes dramatically. First, all the chromatin strands make copies of themselves through the process of DNA replication. They are compressed then to an even greater degree than at interphase, a 10,000-fold compaction into specialized structures for reproduction termed chromosomes. As the cell divides to become two cells, the chromosomes separate, giving each cell a complete copy of the genetic information contained in the chromatin. Nucleolus, again, is a membrane-less organelle within the nucleus that manufactures ribosomes, the cell's protein-producing structures. A nucleus may contain up to 4 nucleoli, but within each species the number of nucleoli is fixed. After a cell divides, a nucleus or nucleolus is formed when chromosomes are brought together into nucleolar organizing regions. During cell division, the nucleolus disappears. The nuclear envelope is the double-layered membrane enclosing the contents of the nucleus. During most of the cell's life cycle, the space between the layers is called perinuclear space, and appears to connect with the rough endoplasmic reticulum. So the space between the two layers of the nuclear envelope The envelope is perforated with tiny holes called nuclear pores. These pores regulate the passage of molecules between nucleus and cytoplasm, permitting some to pass through the membrane but not others. The molecules. The inner surface has a protein lining called nuclear lamina, which binds to chromatin and other nuclear components. During mitosis or cell division, the nuclear envelope disintegrates but reforms as the two cells complete their formation and the chromatin begins to unravel and disperse. Um, so nuclear pores is perforations within the membrane of the nucleus. These regulate passage of molecules, again permitting the molecules to pass through, um, depending on what they are. These molecules are building blocks of DNA and RNA, which are allowed into the nucleus, as well as molecules providing energy for constructing the genetic material. So necessary molecules are allowed. The pores are fully permeable to small molecules up to the size of the smallest proteins, but form a barrier keeping most large molecules out of the nucleus. Some larger proteins, such as histones, are given admittance into the nucleus. Each pore is surrounded by an elaborate protein structure called the nuclear pore complex, which probably selects large molecules for entrance into the nucleus. So again, you have the pores, the nuclear envelope, the nucleolus, and chromatin. Summary: the nuclear envelope is a double membrane composed of outer and inner phospholipid bilayer. Thin space between two layers connects lumen of the rough endoplasmic reticulum, or RER, and the outer layer um, is an extension of the outer surface of the RER. The inner surface of the nuclear envelope has a protein lining called nuclear lamina binding to chromatin and other contents of the nucleus. The entire envelope has numerous nuclear pores or perforations that transport um, molecules. This layer is fully permeable to smaller molecules up to the size of the smallest proteins, but there's a selective barrier against movement of larger molecules. Each pore has an elaborate protein structure surrounding it called nuclear pore complex, which selects molecules for entrance into the nucleus. Nucleotide building blocks of DNA and RNA, as well as adenosine triphosphate, which provides the energy for synthesizing genetic material, also known as ADP, is allowed. Histones and other large proteins also must pass through the pores. These molecules have special amino acid sequences on their surface that signal admittance by the nuclear pore complex. The complexes also regulate the export from nucleus of RNA and subunits of ribosomes. Double-stranded DNA, the definition, is this double strand, a series of nucleotides, and each nucleotide is composed of a sugar phosphate compound attached to one of four nitrogen-containing bases. So, this forms the backbone of the DNA strand. Thank you.